Good morning or good afternoon, everybody. Welcome to today's webinar. My name is Marit and I will moderate today's session about how to generate value out of your data. Before I introduce you to today's speaker, let me quickly introduce you to our company ONTOFORCE. At ONTOFORCE, we enable our customers to make data-driven decisions, drive their innovation, and speed up cycle time. We do so by unlocking and connecting all internal and external data silos, enabling everybody to integrate, analyze, and also extract data much faster. DISQOVER is our self-service linked data platform, which will be further explained in today's session by Philippe Patin. Philippe is our scientific lead at ONTOFORCE. After his explanation and demonstration, you will have the opportunity to ask questions by raising your virtual hand in the Zoom application, or you can type in your question in the chat function. Without any further ado, Philip, the floor is yours.
Thank you, Marit, for the introduction. So good morning, everybody. Good afternoon, everybody, also from my side. So I would like to touch with you today some topics of what our platform can do. You maybe have seen that we already have some other recordings of webinars which are specifically focusing on certain use cases. What I would like to do here today is trying to explain in a high-level way the principles of how we work, how our platform is used, and how we can get to, and that's the first point of the agenda, make data meaningful. I will explain you some concepts which are already for some time around that we try to apply and make available in an attractive product. And then also showing you where it all starts. If you have to bring data together, how we can help you with easing the pain of doing that. And then finally giving you some explanation of how you can use DISQOVER and how you can benefit if you are preparing your data for better linking and harmonization. And then finally, like Marit explains, you will be able to ask some questions if there is a need to intervene there. Not a problem, you can always raise your hand during the discussion even, and already ask your questions so that we can already make notes and prioritize on that. So to start, to make data more meaningful, what does it mean? It's an important principle in our research activities in pharma and life sciences, but also in other domains. And this is a slide which I borrowed from Martin Romacher from Roche. It explains that as an industry, we have an issue with getting grip on our data assets. And so the problem is that more and more data is generated, but less and less we are able to really do something meaningful with that data. It's partially because of the complexity of the data, it's the size of the data, but it's also because we have possibilities to generate much faster, much broader information, and that we, while we are generating data, not think about the future or potential reuse of that data. So how can we start with data, making data more meaningful? If you have a number of different types of data which are buried in different silos, you can have a lot of internal data, could be stored in the cloud or on-premise. You could have external data that you license in, or public data that you use as common knowledge or background information, and you would like to do something meaningful with that to finally get more insights and make better decisions. So what it means is that we try to bring all these sources together to connect the dots. Why? Because people are typically doing specific types of reporting. So if you are in business intelligence or in other applications, you try to get a grip on certain data sources. We try to make this more sustainable in the sense that if we can generate a better graph or network of related data, that the reporting could be very much, let's say, simplified. Instead of doing everything ad hoc over and over again, we try to establish a ground foundation. That graph that we create can also be used for searching. So that means that if you have a general question and you would like to query the system of integrated data sources, you should be able to do that. And on top of that, if you are searching and exploring the data, you should be able to run some proper analytics. It could be in a platform, it could also be in an ecosystem that you are using additional platforms where you send the information to and that you make a kind of a chain of different related applications using the same data. What is the goal of that is, in general, getting insights. Could be that you try to find what you're looking for, quite straightforward. It could also be that you try to find new associations between different data points, or you would like to help this in decision-making finally. If we want to get to this means that we have to start from the beginning with proper annotating, linking, and cleaning the data. It means that we also need an application for power users, expert users, and end users to consume the data, to work with the data. So how does it work in principle? If I take a simple example of a certain source, and I took a few public data sources, you see here a lookup I've done for a certain human gene, CDKN2A, and I get a list of results. This is just the web interface, but you could see this as a table of information. It's very basic, it's only a few columns. There is much more on that system. But what you see immediately is, I'm here for the people who are familiar with UniProt. This is a protein database. You also see a column gene name. It says gene name, but you see immediately also some synonym terms, and actually it's a gene symbol. It's, for example, for Homo sapiens, the gene symbol of that human protein. Proteins are encoded by genes, so there is a certain relationship between proteins and genes. If you would like to make this a little bit more linkable and interoperable and machine readable, we can, instead of just using symbols and just using plain text, we try to encode this with a unique identifier. And so the principle of a URI is a uniform resource identifier, which means that we try to use a standardized, you could also use this as an internationally recognized identifier for the concept I'm talking about. In this case here, I'm talking about human genes. So there is an initiative, what is called the Human Genome Nomenclature Committee, which is officially the instance which decides on the names and the naming and the symbols of genes. So what you have here is that we use the gene symbol, but we also use a prefix. And that prefix is standardized and can be internationally recognized as a identifier for this concept, that specific human gene. If I think about this in a broader way, we can expand this type of information to what we call here the RDF Data Model. RDF stands for Resource Description Framework, and it means that we try to translate a piece of information into a structured representation. So if you see here the column name, gene names, and one concept here, we try to explain this as this identifier has a relationship, that's the meaning of a predicate, with something different, an object. This object is a gene, and again, here we are using a certain prefix. Why? Because there is a certain UniProt here, a certain instance, a certain organization, which has defined exactly what do they mean with a gene. And then the predicates, that's a little bit complex written down here. This is the definition of the relationship between the concept and this concept, gene. It represents, it means as this is of the type that. This is RDF, this is a long text. This is because the standard was already set in almost a little bit more than 20 years ago. So it's already a long time that the semantic web and the principles of the semantic web are already existing. The uptake of that is still ongoing because of the complexity of doing the effort of getting your data in that shape. And that's one of the things we are trying to do. This is like a small sentence of 3 terms. You can also represent this in another way, a little bit more user-friendly or a little bit more readable. So instead of writing down this full predicate explanation with a prefix, you could use this prefix, and there are a number of prefixes which are known abbreviations. So RDF and semicolon is the same as this long stretch. UP or up is UniProt, the abbreviation. This is the prefix for that. So what you can translate or you can represent this information as this symbol is of the type gene. If I go now to a second data source, I have the same thing here. I'm looking here at NCBI gene. And also I did the same search. I searched for CDKN2A, and it was meaning the human symbol, the human gene. So you have the symbol here. You have a list of results. Same thing again. If I'm linking back, I have an identifier, but now I can also come up with other identifiers. There is more than one possibility to represent in a unique way this piece of information. I can use the gene, the gene symbol, like I just did before, or I have 3 ways of using what they call here the NCBI gene ID. This is the official ID, which is used by NCBI gene, and NCBI gene is a kind of golden source for gene information in humans and other species. So there are 3 ways of using that number 1029 in a certain notation with a prefix. That means that there are combinations possible, that not everybody is adopting to the same URIs, but we can solve this by adding some information to that. So again, here you have some prefixes which are added, but now I'm using a certain relationship match. I say this symbol or this URI has an exact match with this URI, and this URI with this URI, etc. And then finally, I can say, okay, this certain identifier is a gene. So what I make here is that this URI is of the type gene again, but now it seems to be another prefix. That's the idea of semantic wrap. It's very simplistic what I'm showing you here, but we try to represent all pieces of information in such a way under the hood of our platform. What is the benefit of that is that we can be very clear in what we represent or what a source is representing, and it can be very easy then to make links. How do we do it? If we take the 2 sources again and the 2 pieces of information, we can add some more information. So I have UniProt again, and I show here a certain symbol, a certain gene symbol is of the type gene. I can say, okay, in another representation of a triple of a subject predicate object, a certain identifier is a protein, and I make a relationship between the 2 concepts. This protein is encoded by this gene. This is what UniProt is saying. I have a number of other prefixes here. So I have my 3 mappings. My symbols are matched with other identifiers, and in addition, I can say, okay, a certain gene is of the type gene, and in addition, my gene or my gene symbol is discussed by a certain reference. People who are familiar with the domain recognize here PubMed, so PubMed IDs. So that means that this gene is somewhere discussed in a certain publication. Cyto is a citation ontology, so it's encoding for relationships between certain pieces of information and references. This is how the data could be represented, and if we really merge that together, we can draw a graph like this. So what does it mean? This concept here, this UniProt ID, is of a certain type protein. It's encoded by a certain other concept, which is a gene. That gene is, like I said, of the type gene, and it has a certain match with other identifiers. And one of these identifiers is also recognized as a gene. And that makes it sometimes a little bit complex. What is now exactly a gene? How do you match all these things together? Because here you have one prefix and here another prefix, but because of the exact matching, we can accept that these concepts are the same thing or meaning the same thing. So it's like twicely recognized as a gene. Fair data. Fair data is an idea about making your data more findable, accessible, interoperable, reusable. It's a concept which was first published a few years ago. It started in our domain, in the life sciences domain, because of the whole evolution and what I was just showing of this semantic web and linked data representation, which was not going far enough to really make your data meaningful, more meaningful. So what they did, the people publishing the fair data principles, they made a number of rules, a number of principles that you can follow to make your data in the first place more findable, explain that your data is better accessible, interoperable, and reusable. And it's from the point of view that your data should be machine readable. So what it means is that if we try to automate processes of bringing data together, doing something at scale with data, so that means not with manual interaction, but with automation, you should try to follow as much as possible the principles. So what it means in the first place, findable means that you should have unique and persistent identifiers. That's what I just showed you with the URIs. You should have basic machine readable descriptive metadata. That's what I was trying to explain very basically with the types. You explain that a certain concept is of a certain type. And then you try to make your data clearly accessible. Accessible doesn't mean that it's open. It means that you explain how machines and humans can get access to the data. That means that you try to be explicit about the authorization, the certain protocols which are needed to get access to, but also the formats which are used. And then the last two ones are a little bit harder sometimes. How to make your data better interoperable. That means interoperability means that you sometimes think about how to link your data to other related data sources. So your machine readable data should be annotated in such a way, and annotation, for example, with a link to PubMed, that you are able to link to other vocabularies or ontologies. What is a vocabulary or an ontology? This is like an official description of information, of concepts, of relationships within a certain niche or subdomain or domain. So interoperability is very important for the next thing also for reusability, because interoperability allows you to link to other data sets and opens up the possibilities for potential reuse. That means that we typically generate data for a specific end goal, for a specific use case, for a specific reason. If your data is more interoperable and findable, accessible, etc., then your data could also be better reusable. And reusable means that your data is not just hidden away somewhere or easy or difficult to reuse because you don't have the correct explanation, you don't have the correct annotation of this data. You try to describe everything well enough so that you can link it to other data sources or use it in other applications. And that makes the data really valuable because sometimes it's very expensive to generate certain data. And that means that you can continue to use the data also for types of use that you didn't have in mind before. So what is also important, and that's something we think about, and I was already mentioning this, is take into account that what we described here is a protein. So really use this as an important piece of information to say, okay, if you would like to search for proteins, this is like a class of information about proteins. Also embrace here the possibilities of, yeah, these are two different classes of genes, but actually they mean the same. So do something with that you capture that all together. And then we have this reference to PubMed. That means if we have PubMed as a data source, this could be an anchor point to get access to PubMed. So my protein and my gene are linked to a certain publication. That means that my publication is indirectly linked to that protein because the gene is directly linked, but the gene is directly linked also with the protein. And that's the way we can work and help you with navigating through the data. That's everything about FAIR data. What is also important here with FAIR data is being pragmatic. Instead of going for the ideal case where you try to make everything fully explained in a linked way as RDF, etc., you try to see where the value gets, where you get value of making your data more fair. Making your data more fair means that you're maybe not generating data from the beginning as FAIR data, that you have to turn it into FAIR data, or that you have to do a certain effort during the generation of your data to make it more fair. And so what does it mean? Making data fair means it's a certain burden that you have. It's a certain effort you have to do before your data is in a more fair shape. By doing this, you invest in time and money in getting your data into that shape. And then you get normally the benefit of that. That means that it's easier to use the data. It's easier to distribute the data in a certain form. It's easier to convert it to certain applications for end users. So end users should normally benefit from that. This is not part of the webinar, but we are also developing a return on investment calculator which can help with making an assessment. What is the effort, if you do a certain effort of making your data more fair, what is the return on investment, what is the return you can generate for your organization. So assume that you do first effort and you get some value out of that. You still need to maintain the data. And then you could decide, and that could be in different iterations, that you say, okay, I will do an additional effort to get more fairness out of this data because of additional use cases. So that's where the principle of fair enough stands for. You try to define the use cases which are important so that you can go in steps in the verification of your data. So you do a second effort. That means an investment in time and money, but then you get really a lot of value out of it because you are broadening the usage. You can use it in a much, let's say, extensive way. Very important here to be realistic on making your data fair. If you go to a final concept, the data UX, the usability and user experience of your data, this is a flow chart of a number of steps which are necessary to get your data into a user interface or an API, which could be consumed by end users or by expert users or power users. So how does it start? You start here in the beginning with defining a use case and you check, do you have the data already? If the data is already existing, is it external data or not? If it's external data, you get to the data. If it's internal data, you try to check, do you have the right access to it? Is there a certain data stewardship plan? Do you take into account that your data should be well preserved, etc.? This is very important. Also, there are a number of UX challenges which could help with easing the pain of doing this. If your data doesn't exist already, you have to generate the data. Could be an experiment, could be other processes where you try to have a proper way of doing your data generation. That means that you're trying to think about the future use of that data, how you have to store it, how do you have to annotate it. Could also be improved with user experience here in the way you are generating the data. And then if you have your data, existing data or newly generated data, it could be in a structured format to a certain extent, could be unstructured, not so easy or a little bit more difficult to work with it. And you also have to maintain some information about what are the mappings, what is the background of information, of knowledge, which is important in your domain. It could be that you tap into existing ontologies or that you would like to manage an ontology yourself. That means that you try to structure the information about the business processes, the research, the way you are organizing your business or your research. And you would like to also manage that. So that's also a challenge to do this in a proper way. So if you have your amount of data, you would like to verify it. That means that you would like to bring it in a shape that you can nicely use it. That verification process is something we can help with. So we can help with, and to a certain extent, in data generation, taking a look to where would you like to go to with your data. So to think about these could be suggestions to generate your data in a better way. Same thing for data stewardship and to a certain extent for ontology management. But the verification process and the integration process is something which is really very well developed inside DISQOVER, our platform. What does it mean is that we can help with generating your data in a more fair state and that we can store it in a knowledge graph. This is our proprietary database system that we have, and that we have the possibility to also create link outs to a big data storage system. So that means that we capture part of the metadata and the data which is important for filtering, for searching, for linking, for browsing, and that we can link out to more raw data sets which are not directly used when you are searching, but maybe that you would like to use or would like to consume in a follow-up step in a downstream process. If you have your data here in an integrated storage system, you can now feed different ways of consuming the data, like using the UI or using an API. How do we get to that? Easing the pain of harmonizing data. I would like to quickly show you, because this is one of the modules we also have in our system, is our data ingestion engine. So what you get is possibilities to do, to a certain extent, manage the data sources, manage the metadata of your data sources, and you can create pipelines, processing pipelines for doing data cleaning and mapping. So what does it mean in practice? You can build a pipeline, you can collaborate on that, and you can run this pipeline on with different schedules. That means that we can tap into the original source. That's the place where the data is generated or initially stored, and that we gradually can pump it through the pipeline. And this is a very fast process. So this is a proprietary process that we have to also embed immediately the principles of linked data and semantic wrap. So if you pump your data, you build your pipeline and you pump your data through your pipeline, you have possibilities also to do proper QC on your data. Why is this important? Because you are sometimes working with millions or even billions of data points, and you would like to keep the control over the processing, or if you have a certain variability in the quality of your data, you would like to monitor that. So we can generate reports which help you with getting an overview of all the data and the data quality. And it can also be used as a reporting tool for and logging for the processes which are running. If you have your data in a more harmonized way, then you can benefit from the principles, what I just explained about fair data and linked data. You can build your own knowledge graph. So what does it mean? You have different data sources, and what I show you here is a screenshot from the public version of DISQOVER. You have different data sources which are all together creating a certain graph, an ontology graph or a data typed graph. All these circles are different data types that we defined. This is a combination of 145 different data sources, public data sources, and you can use this as a resource of integrated data, but you can also represent this here, how this data is all linked together. So what is the interesting thing here? These sources, these public data can be used for data enrichment. That means we have structured already part of the public data. This can be consumed and can be used as a kind of background information, golden source of data to further enrich your own internal data or potentially licensed data. You can just consume this data as is. It's like an ideal world scenario where everybody would take more into account of publishing or generating more fair data. You could easier use it. This is what we already prepared. And we hope that this can be an incentive for people to generate also more fair data, because we try to show you here what the benefits are of easier consumption of that data. So how does it look in detail? And that's something I can also come back to in the demo if necessary. You have here in our data section of the platform a possibility to go to the ontology graph. And if you highlight a certain source, you see that the rest of the graph is grayed out or more yellow, and that you see from a certain data type here, publication, there is like a star of links to other data types. So if you're familiar with PubMed, you know that these are abstracts of publications linked to persons, the authors of publications, also to other data types, which are annotations or tags which are added. If I combine this with a second data source, for example, publications. So if I have, for example, clinical trials, this is the concept of clinical trials. You see that we have a certain graph now, two sets of a graph which are linked together, and that a number of data types are connected. So there are people here who are authors of publications, and there is people here who are, for example, principal investigators in clinical trials. So just switching now to the demo. Please ask your questions already or raise your hand already if there are questions. But what I would like to do here is quickly showing you what the principles are, how we apply it, and how it works in practice. So what I was doing here was showing you how we bring and link the different data sources together. And I can immediately show you how we can benefit from that. So I already showed you this button here, data. Sorry, I'm logged out after a few minutes, so logging in again. This is where we show you how the data is organized. but I'm going back because I showed you that already in the slides. You have here the central pane where you can start your search. And for example, let me do the same thing, CDKN2A, and I'm just hitting enter. So what happens is the system searches across the whole graph and shows you what the results are. You have two types of highlights here. You have a highlight with a background, white background, for example, patents, projects, publications, where you have CDKN2A as a search string and that you see the number of results which are matching here. But you also have two hits with a green background, and for example, one of them is gene. So that means here you have two numbers that there is at least one, and in this case here, 57 hits, which are semantic hits to concepts. So CDKN2A is an official label or a synonym term for 57 different genes in different species. So this is the distinction you can make. This is also the possibility to make direct choices to go to specific data types. So assume that I would like to search in a broader way. For example, CDKN2A is a certain gene symbol, but if I click on that, I can take a look to all the synonym terms we have. And so what you get here is that CDKN2A is recognized as a human gene, but also a lot of other meanings because all these species have CDKN2A as official gene symbol. But CDKN2A has other names which are also used, like P16 or ARF or INC4A. These are other names for the gene products. So I could expand my search if I think that's relevant and search in a broader way. That gives me the possibility to really actively make a choice for a certain concept and the synonyms and expand my search. If you remember the number of hits I had here in the beginning, 6,000 and a bit. If I'm taking the synonyms into account, I have many more hits. What does that end up for me? If I click here, I can go to publications. And what I've done here is, or what is done here in the public version of DISQOVER is showing you an overview with a dashboard of all the publications which are hits. On the right side, you have a lists view where you see the different publications. So this is an endless scrolling list where you can go to the different publications, but I still have 30,000 publications. So what I could do is, for example, actively search for recent publications and, for example, take a look to related data or make additional filterings. For example, I would like to combine this with, I'm just making a term, infection. So I'm making a certain selection because I'm interested in infection as a term and my gene symbol. I can add here different terms, and you see a reduction in the number of search results. You see where the publications are published, in which journal. You see certain descriptors, and if you want, you can add your own facets to this dashboard. Let's say that I'm interested in only taking a look at review articles. I would like to see more summary papers. So I click here on this symbol, and I see all the different properties that are available for publication. And I take here publication type by selecting this. And if you select multiple, you will have other possibilities here to filter or to do analytics. I would like to have a certain filter, and I would like to show it as a list. So show this as a list and say, okay, show me review articles. And I would like to see who are the top authors in certain review articles. It seems to be that there are no real people pointing out. So maybe I can relax my filter a little bit and say, okay, I will take a broader section of the domain and take a few years more. So here I see a number of top authors popping up. I'm able to make selections. For example, I can select the publications of this person and I can take a look. What it means is that we are looking here to the details of one concept, so one publication in this case. If I click here on this toggle, I can see the different data sources. In this case, it's mostly PubMed related data, and there are some links created to ORCID if you're familiar with that. This is a repository of profiles of scientists mainly who are actively attaching publications to their profile. So this can help to resolve the names of people and to turn them into physical people. So these are the details. I also have a link to the external application. This is something we try to provide everywhere possible. So that means that if I follow this link, that I go to the original publication, or at least submission in PubMed, like you see. Or if I'm following here the keywords, I'm pivoting around the term. That means that I can go to everything which is linked to that, and that this is linked to data at work. If I'm interested in lymphatic metastasis, if I click on that, I will go to that concept. So let me just do this to show you. This is the concept lymphatic metastasis, and I see everything related to that. A high number of publications, a number of pathways, a number of genes, a number of disease-gene relationships, etc. But I'm not directly interested in that. I'm going back to my publications. This is the possibility I have here to go back to my different tabs. So what is important here? I'm looking to publications, but actually I want to go back and take a look to the genes. So if I'm just clicking on this, I can see my initial search results. And if I just have to look here, yeah, gene, I'm going to gene, and I can take all human genes. So I'm filtering for the organism Homo sapiens, and I see here the different human genes. The first hit is the exact match. The other hits are hits where the term CDKN2A, and you see still the highlighting, is somewhere in the text. So if I click on the gene I'm interested in, I see the information coming here from this gene. It's a kind of a summary report with all the details. And if I click here on data sources, you will see that a lot of data sources are presenting information. The ones with a colored background are really presenting data. The ones with a white background are providing links, or at least links. What is also now important is that this is like a unified concept. And if I click here on this term, you see, on this symbol, you'll see that we have different URIs brought together. So these are the URIs that we use in DISQOVER to bring pieces of information from different sources all together. Because if a source is referring to a certain gene, we try to map it to a URI. And every source is using another combination of URIs, but we know from certain sources or ontologies that certain URIs are similar to each other or are meaning an exact match. So this is what we have here. This is the application with all the possibilities that you can navigate and link and create a search overview. What is now interesting here is that I can easily create my own dashboard. So I have a number of genes here, and I can say, okay, I would like to link from all these genes. I would like to make a link to pathways. So I'm linking to pathways, and I see all these genes, how they are linked to different pathways. This gives me the possibility to do an investigation on, let me say, I only would like to see human pathways, and there are a number of other possibilities available to filter, but I miss something because I would like to see a representation of the different genes and proteins which are related in this pathway and which are potentially associated to certain diseases. So what I will do is I will further narrow down because the list is quite high, so it will not be so relevant to go into detail, to link into detail. So I will go back and not take all the genes, but take the single gene and go to the related pathways. Just taking a look here, 47 pathways. And I would like to see, so this is a smaller list of potential hits. I would like to see, can I link the associated disease of my pathways to certain genes. And I would like to analyze this with a Sankey diagram. So what happens is it shows you the top genes which are linked to the top pathways, to the top diseases. And now I can also magnify this in a full screen mode and see here, okay, I see certain associations continuously popping up. I can make a link, for example, of CDKN2A, and I can actively filter for that and just show me CDKN2A, how they are related to, how the pathways which are CDKN2A related, related to all these diseases. I can go back to my pathway view and continue if I want. I can follow different links, I can go back, but what I have here is an overview of different searches. If I click on this button here, I can see what I've done before. So I started with searching for a gene and I went to pathways. I went back from a single gene to the pathways, and I also took a look to publications. This is, in a nutshell, the capabilities of the platform. So what it means is you can, by using this data ingestion engine, streamline your data harmonization process, preparing your data for the user interface. I haven't shown you anything about the API, but every search you can do here can also be done with the API. You can build dashboards if you have the right credentials. You can build dashboards for colleagues. You can, as a person, an individual person, create your own dashboards. There is a possibility to download the data if you want, to consume the data again. There are possibilities to link out to other applications. What I'm showing you here is the public platform, public DISQOVER platform. The real implementation is that this installation can be installed locally in your private cloud or on premise. You can load your own data. This is not possible with the public version of DISQOVER. You can load your own data and you can consume and manipulate the data in the way you want to make it ready for searching and navigation. So this was it. If there are any questions, you can raise your hands, and I think Marit will try to coordinate the different questions.
Yes.
Thanks for your attendance.
Thank you very much, Philip. There was already one question asked.
Mm-hmm.
Let me check. Yes. Is there a limitation to the number of data sources that the DISQOVER platform can handle?
Yeah, it depends a little bit on the type of data you are using. In a sense, there is no real limitation. We can handle a lot of data. If you're a little bit familiar with sizes of information, we can handle at the moment, or we have done experiments with more than 10 billion different data points. So to answer that question, it's the combination of how big the data is, how complex it's interlinked. But for the moment, we haven't seen any limitations so far. We also have still possibilities to scale with the hardware.
Okay, great. Thank you.
Other questions?
Yes, there's another question coming up. Can the DISQOVER platform be used for applications outside of, let's say, research and clinical?
Yes. What you have seen here during the demo is some information about publications, clinical trials, etc. But it's actually the way you try to model the data. It could be that you also are interested in data which is more involved in monitoring or in adverse events reporting or in other parts of life science business. So it depends on the data you ingest. You could see it actually as a kind of an empty box, the platform where you can feed the data you want. We have a current focus on life sciences and healthcare, but we also have a few projects outside that business.
Okay,
Other questions?
I don't see any new questions coming up. If so, please put them in the chat or raise your hand so I can unmute you. No, I think everything was clear, Philip. So thank you very much for explaining. Thank you everybody for attending today's webinar. If there are any questions whatsoever, or you want an explanation on more specific use cases, please feel free to contact us to get an offline meeting with us. But for now, I think we can wrap up the session for today. So thank you everybody for attending the session. Stay safe in these crazy times, and hope to see you next time. Thank you. Bye-bye.
Bye-bye.